Structure of bacterium. Structure of bacterium. A bacterium mainly consists surface adherents, cell wall, surface appendages, protoplasm. The structure of a bacterium mainly consists of 4 parts: surface adherents, cell wall, surface appendages, protoplasm. Surface adherents, that is glycocalyx. Surface appendages, flagellum, pili, fimbrium. Surface appendages are the flagellum, pili, fimbriae. We will discuss that in detail each one. Now first come to the surface adherents. That is glycocalyx. Some bacteria, not all bacteria, enveloped by a coat that is called glycocalyx. It is cell wall of bacterium. Outside the cell wall, a cover that is glycocalyx. Some bacteria enveloped by glycocalyx. Glycocalyx composed with polysaccharides mainly and proteins also present. Glycocalyx composed mainly with polysaccharide and some amount of proteins also present. The main function of glycocalyx, attachment. Main function of glycocalyx is attachment. Helps to bacterium to attach host tissues or to the substratum in aquatic environment. Glycocalyx main function, attachment. It helps to bacterium to attach either to the host tissues or to the substratum present in aquatic environment. Based on how it attaches to the cell wall, it is the 2 types. If the glycocalyx loosely attached to the cell wall, called slime layer. Glycocalyx loosely attached to the cell wall, slime layer. If it is tightly attached to the cell wall, called capsule. Glycocalyx present outside the cell wall, in some bacteria, not in all. The main function is the attachment. If glycocalyx loosely attached and easily washable, called slime layer. Function of slime layer, besides the attachment, slime layer protects the cell from loss of water and nutrients. Slime layer protects the bacterial cell from loss of water and nutrients. If this glycocalyx tightly attached to the cell wall, it is called capsule. Besides attachment, capsule also helps to bacterium from desiccation. Capsule protects bacterial cell from desiccation. But the common function of the both slime layer and the capsule collectively called as the glycocalyx, that is the attachment. Besides attachment, they perform other functions also. And next one, cell wall. Cell wall of bacterium. Bacterial cell wall not composed with cellulose. Cellulose is completely absent because it is the prokaryotic cell. Bacterium is the prokaryotic cell. Cellulose is completely absent. Instead of cellulose present, the bacterial cell wall composed with peptidoglycans, also called mucopeptides. Peptidoglycans are mucopeptides. Cell wall not consists cellulose. Cellulose is completely absent. So in all prokaryote, the cell wall not consists the cellulose. Bacteria is the prokaryote. So in this cell wall also cellulose is absent. Instead of cellulose present, peptidoglycans or mucopeptides. These peptidoglycans consist N-acetyl glucosamine, N-acetyl muramic acid molecules arranged alternately, NAG, NAM, N-acetyl glucosamine and N-acetyl muramic acid arranged alternately. These two are amino sugars. N-acetyl glucosamine and N-acetyl muramic acid are amino sugars. They are amino sugars. That means they are heteropolysaccharides. Heteropolysaccharides. Now, at one side to the muramic acid present peptide, a small group of amino acids attached to one side to the muramic acid of NAM, N-acetyl muramic acid. So it is the structure of the cell wall, structure of bacterial cell wall, the chemical composition. Now, function of bacterial cell wall, it maintain particular shape to the cell. It maintain particular shape to the cell. It protects the bacterium from collapse and burst. When water that enters into the cell, that cause pressure on the cell wall. So the cell swell and finally it may burst. Cell wall that protects the bacterium from that bursting. When water that completely comes out, cell may shrink. Here also cell wall that protects from cell from that collapsing. That means cell wall protects cell from osmotic lysis. Cell wall protects cell from osmotic lysis. Some cell wall substances cause pathogenic nature. Some cell wall substances cause pathogenic nature to the other organisms. Christian Graham classified bacteria into two types based on staining procedure. That stain mainly absorbed by the cell wall. Scientists classified bacteria into two types based on staining procedure. Scientists classified bacteria into two types based on staining procedure. Christian Graham. Stain used by him. Crystal violet. Stain used by him is crystal violet. And that procedure called Graham's staining procedure. What is that procedure here? Bacteria, procedure of staining, gram staining procedure. Bacteria taken from culture. Taken from culture. What is the meaning of culture here? One bacterium placed on the nutrient medium that multiplies in many ways. Multiplies and more number of bacteria present. All bacteria are the same kind. So, in a culture, only one type of bacteria present. That's the reason he taken that bacteria from the culture. Next. Heat fixation. Bacteria taken on the slide, and when focused to the heat or the spirit lamp, they are attached to the slide. Treated with crystal violet, all bacteria stain, that is color either purple or violet. Next, it dipped in iodine solution. Then treated with ethanol or acetone. If bacteria lose color, they call gram negative. If bacteria retain color, they are gram positive bacteria. Gram staining procedure. First, he taken bacteria from culture, exposed to the temperature, so all bacteria attached to the slide, that is called heat fixation. Next, all bacteria are treated with crystal violet. All bacteria change their color, that means all they stain. The color is either purple or violet, and then that slide or the bacteria treated with iodine solution, and then all the bacteria are treated with either acetone or ethanol. When bacteria lose color, they call gram negative bacteria. Example, lactobacillus. If they retain color, they are called gram positive bacteria. Gram negative bacteria. Example, Escherichia coli. Gram negative bacteria. Example, lactobacillus. Simple procedure. By the simple procedure, he classified, Christian Gram classified bacteria in two types, gram positive and gram negative. A question that arises, why gram negative bacteria lose color when treated with either ethanol or acetone, and why the gram positive bacterium retain that color? In case of gram positive bacterium, the cell wall consists teichoic acid. Teichoic acid present in gram positive bacterial cell wall. It holds stain particles. Even treated with the ethanol or acetone, never lose that color. But the type of teichoic acid is absent in gram negative bacteria. So, in gram negative bacteria, the cell wall not holds the stain particles. So, when treated with acetone or ethanol, it loses that color or the stain particles easily. It is a gram staining procedure. That's about the cell wall. And the next one, surface appendages. Surface appendages. They are the flagella, pili and fimbriae. Surface appendages. Three types: flagellum, pili and fimbriae. Type of protein present in them: flagellum. Bacterial flagellum consists flagellin protein. Pili composed with pilin. Fimbriae composed with fimbrillin protein. So, these are the three different structures present in the surface appendages, composed with three different types of proteins: flagellum composed with the protein flagellin, pili composed with pilin, fimbriae composed with fimbrillin protein. And function: flagella helps in locomotion of the bacterium or the movement of the bacterium. Both pili and fimbriae helps in attachment. Pili and fimbriae helps in attachment. Of course, the pili also helps in conjugation also. But common function, that is the pili and fimbriae attachment, helps to bacterium to attach host tissues or to the substratum in aquatic environment. So, the structures of bacteria helps to bacterium to attach to the host tissues or to the substratum in aquatic environment. Both glycocalyx, pili and fimbriae. The three structures, what are those? Glycocalyx, pili and fimbriae helps to bacterium for attachment. Now, first come to the flagellum. Bacterial flagellum. Differ from eukaryotic flagellum in bacterial flagellum differ from eukaryotic flagellum. Bacterium is the prokaryote. That means the prokaryotic flagellum differ from eukaryotic flagellum in absence of 9+2 configuration. Here, the type of configuration we observe in eukaryotic flagellum: presence of 9+2 configuration. In case of the eukaryotic flagellum, peripherally present 9 pairs of microtubules, centrally located two single tubules. That configuration is called 9+2 configuration. That type of arrangement is absent in case of the prokaryotic flagellum or bacterial flagellum. Composed with flagellin protein. Here, eukaryotic flagellum composed with tubulin protein. Tubulin protein. Eukaryotic flagellum composed with tubulin protein, bacterial flagellum composed with flagellin. Not covered by plasma membrane. Covered by plasma membrane. Eukaryotic flagellum enveloped by plasma membrane, but prokaryotic flagellum or bacterial flagellum not covered by plasma membrane. These are the main difference observed in between the bacterial flagellum and eukaryotic flagellum, but the function that is the common. In both bacteria and eukaryotes, the flagellum helps in the locomotion. Surface appendages, flagella, pili and fimbriae. All these surface appendages arise from plasma membrane, cell wall, and it is the plasma membrane. Either flagella or fimbriae or pili. All these three surface appendages arise from plasma membrane. Now come to the flagellum. Already we discussed about the differences between the eukaryotic flagellum and prokaryotic flagellum, that is the bacterial flagellum and eukaryotic flagellum. Now, flagellum in bacteria consists mainly three parts. Flagellum in bacterium consists mainly three parts. Cell wall. It is the cell wall. The plasma membrane, Now, part of flagellum embedded in plasma membrane, basal granule. From this, a hook is formed. That hook ruptures cell wall. It ruptures the cell wall. So, it is the hook. From this hook, long slender structure that arises, it is the filament. So, bacterial flagellum consists mainly three parts: basal granule embedded in plasma membrane, hardened and rigid structure, that is the hook that present in or embedded in cell wall, and long slender outside the cell wall structure, that is a filament. Only filament that show rotatory movement. By rotatory movement, bacterium that show locomotion. Movement showed by flagellum, that is rotatory movement, and especially the filament part of flagellum that show rotatory movement, and flagellum that moves from one place to other place. Next one, pili. They are less in number. They are shorter than filament but longer than fimbriae. They are hollow structures. Already we discussed about define that pili and fimbriae helps in attachment of bacterium to the host tissues or to the substratum in aquatic environment. And another function pili that perform attached to the other bacterium during conjugation. That pili is called sex pili. Besides attachment to the host tissues or to the substratum in aquatic environment, pili also helps in conjugation, and it produces the conjugation tube between two conjugates. We will discuss that in the sexual reproduction. Fimbriae, these are bristle-like structures mainly meant for attachment.